Welcome to today's Perspectives in Biology seminar. Perspectives in Biology is a seminar series hosted by the Biology Department of the University of Michigan-Flint. I'm Jill Slater, a lecturer in the department, and it's my privilege today to introduce our speaker. My colleague, Dr. Jill Witt, will be handling the question and answer time at the end of the talk. Please feel free to use the Q&A box to type in questions at any time during the talk, and our speaker will address them at the end. Today, we are very pleased to welcome Dr. Ariangela Kozik. Dr. Kozik earned her BS in biotechnology from Calvin College and her doctorate in comparative pathobiology from Purdue University. She is currently a postdoctoral research fellow at the University of Michigan, Ann Arbor, in the Division of Pulmonary and Critical Care Medicine, where she studies the respiratory microbiome in adult asthma. Dr. Kozik is also an advocate for institutional equity and inclusion, serving as the president of the Medical School Postdoctoral Senate and the chair of the Postdoctoral Family Association. She is the co-founder and vice president of the Black Microbiologists Association, also known as Black in Microbiology, a new organization committed to supporting the careers and professional development of Black microbiologists across the country. Today, Dr. Kozik will be speaking to us about fantastic microbes and where to find them. Welcome, Dr. Kozik.
Thank you for that introduction, and thank you for having me today. So today I'm going to talk a little bit about myself and my path to microbiome research, and I'll tell you about my work, and then you'll get a chance to ask me any questions that you may have. So first, I'm going to talk about the microbiome. So my first love is microbiology, which is a relatively old field of study. Microorganisms are everywhere. They inhabit nearly every ecosystem on the earth. We are made up of more microbial cells and genes than human cells and genes, and we cannot live without them. I'm a microbiome scientist. A microbiome is a collection of microorganisms, so bacteria, viruses, fungi, and their genes that inhabit a particular environment. There are scientists who study the microbiome of almost every ecosystem you can think of. So the soil, ice in the Arctic, hydrothermal vents at the bottom of the oceans, animals, plants, and of course humans. Microbes are essential for life. We depend on them in ways that you may not have considered before. They generate oxygen, they recycle the nutrients in our soils, which we need to grow food. They help us make other foods, so bread, cheese, wine, kimchi, other kinds of fermented foods. They all require microbes. With the help of rapidly advancing technology, we can now study the microbiomes of many different environments. And the more that we learn, the more we come to understand just how critical the various microbiomes on our planet are. So I study the human microbiome, specifically in the gut and the lungs. If you're like me, you grew up thinking about microbes, especially bacteria and viruses, as germs that were bad for you and that cause disease. To be fair, many of them are quite unpleasant and lead to things like food poisoning, cholera, influenza, and now COVID-19. But we also know that there are many non-pathogenic microorganisms normally associated with nearly every body surface. We know that these microbes help us digest our food, adjust to our environment, they protect us from unpleasant illnesses and infections, but we don't yet know the specifics about how all of that works. So humans are actually made up of an equal number of microorganisms in human cells. When it comes to the number of genes, which are the sets of instructions that organisms follow to make proteins, we are vastly outnumbered by the genes that are contributed by our microbes. We now know that the presence of these organisms is important for our health, and we think that the microbiome has an important role to play in many diseases such as diabetes, rheumatoid arthritis, asthma, allergies, and even depression. When people think about the microbiome, they usually mean bacteria, likely because a lot of our technologies have been developed for the study of bacteria specifically. I'm going to be focused on bacteria for this talk as well, but it's important to know that your microbiome is more than just bacteria. There are also viruses, there are fungi, there are organisms called archaea, and in some cases even parasites that are associated with our bodies. Your microbiome is unique to you, and it responds to your environment and your exposures throughout your lifetime. We're only just beginning to understand the details about how this works, but this information has resulted in a shift in the way that we think about medicine. One of the big biomedical challenges we face today is figuring out how the microbiome is involved in human health and disease. As I stated, the microbiome is the collection of organisms and their genes that live in, on, and around us, and it's been implicated in several diseases and disorders. Microbiome research is a rapidly developing field, but it has become clear that in order to fully realize the nature and the potential of the human microbiome, we need to recognize it as an ecosystem that is complex, dynamic, and responsive to its environment. The rapid pace of emerging sequencing technologies is matched by advances in computational methods that can allow the assessment of interactions within communities of microbes and between microbes and the host. The hope is that eventually, by understanding this part of our world, we can direct that knowledge into improving our health. Before I talk about my research in this area, I want to back up and tell you a bit about how I got here. So this is my path to microbiome research. I'm originally from Indiana, but the part of Indiana that sort of belongs to Chicago on the coast of Lake Michigan. Back home, we call it Chicagoland. I'm the daughter of an educator and an electrician. I've always been curious, bursting with questions and wanting to know things. As a child, I would often be doing kitchen experiments at my grandparents' house while my parents were working. This involved me going through the cabinets and mixing up stuff to see what happens. My grandparents were never amused or entertained by this, but it kept me entertained. When I started school, I loved it. I became interested in science at an early age, starting with collecting rocks and doing science projects. I discovered microbiology when my parents bought me my first microscope for my 8th birthday. My mom showed me how to make slides to look at things under the microscope, and I was hooked after that. In school, I was very involved in like extracurricular activities and things like Science Olympiad. I'm a musician, so I was also in orchestra and marching band and things like that as well. After high school, I knew that I wanted to do research, but not what kind of research I wanted to do. I set off for college intending to study neuropsychology. I was really interested in human behavior and why people do the things they do and how that was related to the brain. But I went to Calvin, which is over in Grand Rapids, Michigan. I ended up majoring in biotechnology. After undergrad, I earned a PhD from Purdue and accepted my current position as a research fellow at U of M in Arbor. But let me back up and explain some details there. My freshman year at Calvin, I was invited to apply to this new course that was being taught to first-year biology students. The course focused on finding new bacteriophages. Bacteriophages are small viruses that can infect and kill bacteria. There are 10 million trillion more of them than any other organism on the earth, including bacteria combined. They are found everywhere that bacteria are. Each type can infect a specific type of bacteria in order to replicate, and they can do this without harming other cells and other organisms. So because of that, they have the potential to be an alternative to antibiotics. So there's a lot of interest among the scientific community to invest in phage research. So the phage course was an outgrowth of that investment. It's a 2-semester program where students isolate phage from various environments, very likely finding phage that have never been seen before. And the class spends the remainder of the first semester purifying and characterizing the phage that they found and evaluating phage diversity in the environment. We're able to visualize the viruses via electron microscopy. And in the second semester, the students use bioinformatics and comparative genomics to analyze and annotate the phage genome and compare it to other phages, which contributes valuable insight into the potential form and function of the virus and adds to the body of scientific knowledge. So this course was super cool to me because research methods and laboratory skills that we were taught in a hands-on setting while I learned to characterize and analyze phage. And then we got to catalog our data in public databases. And so we were making a real contribution to scientific knowledge. So after a lot of environmental sampling, I have a picture of that on the left here on the slide. I finally got evidence that I had captured a phage in my sample, which you can see in the plate. Those little gray circle areas means that that's an area where the virus had eaten the bacteria that I had plated on that plate. So I was super excited to have finally gotten something. And so it turns out that the phage that I found was one that had been not seen before. And so I was able to name it. I named it Mycobacteriophagianaea. Mycobacteria is the type of bacteria that particular virus was infective against. I was able to analyze it and annotate its genome and then publish that information as part of scientific knowledge. So I spent the rest of undergrad isolating bacteria and looking for phage in termite gut samples, working on insect, environmental, microbiome questions. So I was really hooked on microbiology and microbiome research after that. I knew that I wanted to pursue microbiome research as a career path. As a side note, in 2017, we were actually, not we personally, but scientists in California were able to treat a man who was dying from a multi-drug resistant infection. They saved his life by being able to give him a phage therapy. So this area of research is currently booming with potential to develop alternatives to antibiotics to potentially save lives for people who get drug-resistant infections. So that's really cool. After undergrad, I decided to pursue a PhD so that I could be a research scientist. I was really interested in the microbiome, like I mentioned. I wanted to know about ways to study the microbiome and bioinformatics. For my graduate work, I conducted research on a specific inflammatory signal to find out what happens to the gut microbiome during inflammatory bowel disease when that particular signal is removed. Many of the techniques and skills I use in my work today, I learned during graduate school. I also had my first child during my graduate training and founded the Graduate Parent Support Network at Purdue. It's a first of its kind organization to support graduate students with children during their graduate training. It's also those are two really hard things in isolation, but especially when you combine them. And so I wanted to ensure that there was access to resources that we need to be successful during our graduate training. That brings me to my current position today. So after grad school, I determined that I really wanted to do microbiome research in a clinical setting. I was really interested in direct human application. I wanted to work on understanding how the human microbiome is involved in health and disease. After checking out several labs and projects at a variety of institutions, I found my current lab at U of M and Arbor. I was really excited about my PI, Dr. Huang's, her work with asthma and COPD, and I saw a lot of potential for me to continue to grow my skill set there. So that's where I landed. And so now I'll tell you about some of the work that I do there. So in the conventional practice of medicine, a certain diagnosis may have any number of drugs that have been approved as treatments. This treatment is generally given to everybody with the diagnosis without regard to the individual characteristics that may influence its effectiveness. As a result, that treatment may work for some patients, but not others. Some people may experience side effects, others may not. The group for whom the treatment isn't tolerated or doesn't work is left to find other options, and those options may not be affordable and they may not even exist. One disease where we're seeing this a lot is asthma. So asthma is a chronic disease primarily featuring airway inflammation. It is one of the most common diseases in the United States. Persons with asthma experience a sustained increase in airway inflammation that is further pronounced during an exacerbation, or what many refer to as an asthma attack, is a term you may have heard. Sometimes asthma attacks can be so severe that they lead to hospitalization or death. Known factors that seem to initiate attacks are called triggers. Triggers can vary from person to person, and they can include things like pollutants or pests and even stress. Asthma is burdensome on both the patients and the communities, especially in poor and marginalized groups. There's mounting evidence of disparities in prevalence, risk factors, access to care, and morbidity. The primary long-term treatment right now for asthma is inhaled corticosteroids, which I will refer to as ICS. They aim to prevent the swelling that leads to the symptoms. In most people, ICS does dramatically reduce the swelling, but there are some patients for whom ICS therapy is partially or completely ineffective, and we have no idea why that is. And we also don't know why the symptoms and triggers vary from person to person. So clinicians have long noted the wide variations in symptoms, disease course, triggers, and treatment response among their asthma patients. Over time, it became clear that an asthma diagnosis can cover a wide range of clinical characteristics. For example, there's severe asthma, there's allergic asthma. This is the type that most people are familiar with, but then there's asthma that appears to be associated with obesity, with smoking, with exercise, and other different kinds of factors. And so we now understand asthma to be comprised of phenotypes, which is a recognizable cluster of demographic, clinical, or pathophysiological characteristics. So right now, one of the only ways we have to differentiate between the types of asthma in the clinic is based on the presence or absence of what we call T2 inflammation. T2 inflammation is driven by immune cells that are called eosinophils and specific inflammatory signals called cytokines that are called IL-4, IL-5, and IL-13. In the clinic, we currently measure the levels of eosinophils in the blood or sputum, which is mucus from the airway, to classify patients. A level equal to or greater than 2% of eosinophils is considered T2 high. Sometimes the levels of IL-4, 5, and 13 are also assessed. We know the most about allergic asthma, which is common. It's classified as T2 high. It typically starts in childhood and it's linked to allergies. This is the classical asthma symptoms and pattern of disease that most people think of when you think of asthma is allergic asthma. When it comes to type 2 low asthma, though, where you don't have the predominance of eosinophils and type 2 cytokines, there's a lot that we don't know. The symptoms range from mild to severe in each type, both type 2 high and low. Typically, ICS treatments work for most people that have T2 high asthma, but people with T2 low asthma are more likely to be non-responsive to ICS. Despite the differences in the clinical and the biological features, we still lack precise biomarkers that we can use to pinpoint the underlying mechanism behind the patient's symptoms. So we need more precise diagnostic and therapeutic options so we can better serve the patient population. So that brings us to my work on the airway microbiome in asthma. So when most people hear microbiome, they think of the gut, but we are interested in the respiratory system. As recently as 2010, the conventional wisdom was that healthy lungs were free from bacteria. This was based on the fact that we've been trying to culture bacteria from lung tissue for over 100 years without very much success. So people just assumed that meant that, well, there's no bacteria in the lung. However, the development of culture independent methods has shown that healthy lungs indeed have their own microbiome. The respiratory tract provides an interesting niche if we think about it. It's ecologically very different from the gut. There are minimal physical barriers between the larynx and the deepest parts of the lung. So the tract from the oral cavity down to the lung, it's pretty much free and clear there. The microbes and things can move. There aren't a lot of physical barriers and sphincters like you have in the gastrointestinal tract. But there's also constant contact with the external environment, so the outside world, via inhalation and microaspiration. A microaspiration is where you droplets from your oral cavity make it deep into your airway. This happens normally in just the course of breathing and talking and eating and things. So there's actual crossover of materials from your oral cavity into your airway. So we introduce microbes to our respiratory tract that way. There's also a temperature gradient. So it's cooler at the mouth where it's closer to room temperature to warmer deep in the lungs as we move towards core body temperature. It's also a moist environment. There's mucus and other moisture that's constantly happening in your respiratory tract. So there's bidirectional movement of mucus. Inhaled substances that we breathe in from the air and microorganisms are constantly going in and out with each breath. So the upper airway, which is the part that's in your head, generally has a higher bacterial density than the lower airway, which is the part that's in your chest. But there's compelling evidence for differences in the airway microbiome associated with airway inflammation and with asthma, which makes sense if you think about the kinds of environmental conditions that you have in the respiratory tract to support microorganisms. So previously, asthma research has been focused on studying two main layers of information to understand asthma. First and foremost, the clinical features, and then host features like innate and adaptive immune cells and immunity, genetic risk factors, and airway epithelial biology. These two pieces are certainly closely connected. However, our lab at Michigan Medicine is interested in a newly appreciated piece of the puzzle, which is the microbiome. We know that the microbiome is linked to processes of host immunology and of genetics and the biology of epithelial layers, and so we suspect that it plays an underappreciated role in the clinical manifestations of these phenotypes. Moreover, the microbiome itself may impact or be impacted by a number of important host and lifestyle factors that may also be related to asthma. For example, we have environmental exposures and other health conditions. We think that by leveraging multi-omic data, so genomic, proteomic, understanding layers of proteins, metabolites, what's being made, what inflammatory signals there are, by combining this data, we can reveal which different facets of the processes. We can start to piece together the connections between the microbiome, the host, and the clinical manifestations of disease. So our lab is focused on the application of these concepts in the study of both asthma and chronic obstructive pulmonary disease, which is also referred to as COPD. So thankfully, technological advances in microbiology and data science are moving us in a different direction from the conventional understanding of pathogenesis and treatment that I mentioned earlier. So in the case of microbe-mediated diseases where we suspect that the microbiome may be involved, we now know that we can take patient data, which can be anything from clinical symptoms to test results, activity surveys, allergy tests, like blood profiles, things of that nature. And then we can combine that with microbiome data from a person or populations of people. And then we can use computers to look for subgroups or clusters of patients with similar disease features in microbial communities. And that eventually might help us develop and determine the best course of treatment for them. In this way, we can also research the underlying biological processes that are involved in a disease and better understand how it impacts different groups of people. So therefore, the goal of our clinical studies is to build very well characterized adult asthma cohorts that can be used to ask questions that link the clinical features with the multiomics data and the host biology. With that in mind, we collect a wide variety of information from each of these layers. Our subjects undergo baseline clinical characterization that includes detailed histories, lung function and allergy testing, and nutritional profiling. To capture different facets of the microbiome, we collect lots of different samples, including oral wash, induced sputum, lower airway samples, and stool samples. We employ 16S rRNA gene sequencing and metagenomic sequencing methods to characterize the microbiome. And then to gain insight into host biology, we characterize the blood and sputum immune cells and collect blood and sputum cytokine profiles. We also have blood and urine samples from metabolomics. So we collect really a wide variety of data so that we can put as big of a picture as we can to what's going on in each individual subject. So a couple of key differences between what I've talked about and what we already know from the literature, and what we are equipped to ask of the subset of participants that our study population is currently enriched for. But right now we have a lot of mild to moderate asthmatics in our study, but a lot of our existing knowledge comes from severe asthma, where it's clearer to see, okay, this is associated with like severe disease, and these are the folks that don't have asthma at all. So those are really big contrasts. So it's easier to see large differences there. But we know less about what's going on in mild to moderate disease, where like someone definitely has asthma, but they don't have as severe as a response or severe of clinical characteristics as people who have severe asthma do. But that doesn't mean that that patient group is not important, because we still need to understand what's going on in each stage of the disease. And so our study is focused on what is going on in mild to moderate disease. So I mentioned induced sputum as a sample type that we work with, but I want to talk a little bit more about that, about what that is and why we use it. So there are three major methods of collection for airway samples to assess inflammatory cells in asthma patients. These methods are also used for microbiome profiling. The least invasive is induced sputum, which that involves breathing in an aerosolized saline solution and then coughing the sputum or the mucus from your airway out into a cup. So the cells are then counted microscopically. The major drawback is that the exact source of the cells, so like what level of the airway those cells come from, is not readily known. Bronchoscopy, which is more invasive of a procedure. It's a more invasive technique and it requires some sedation for the subjects. Both the two techniques on the middle and the right, bronchoalveolar lavage, which targets cell collection from alveoli within a specific lung lobe of the lung. And then there's also endobronchial biopsy, which targets a specific part of the airway wall. Those are two different sample collection methods we can use if someone undergoes a bronchoscopy. So bronchial alveolar lavage yields an inflammatory cell count similar to what we can get from induced sputum. Endobronchial biopsy allows for inflammatory cell count within different structural compartments in the airway wall, so we can look at different levels of the airway. It also allows for some analysis of the airway structure, including the epithelium and the smooth muscle cells. So so all three methods are important and give us important things, but the last two they're just very invasive. And so induced sputum is relatively cheap and it's a non-invasive procedure. It's useful for microbiome studies in asthma cohorts because profiles from this sample from work from my lab, other people in my lab have shown that this sample type, induced sputum, does give us microbiome readouts that are similar to the more invasive lower airway samples. So that's what that is and why we use it. So I'm going to talk a little bit about one of my projects today, focused on one of the types of asthma. Specifically, that's going to be obesity-associated asthma. So obesity is characterized by increased body adipose tissue and chronic systemic inflammation. So adipose tissue is made up of adipocytes, which store energy in the form of fat, and they can contain blood vessels and nerves. We now know that adipose tissue is very active in the body, which is pretty amazing. In addition to sensing and responding to the body's energy needs, it also secretes a number of hormones that can impact other tissues in completely different areas of the body. So immune cells also play an active role in maintaining the hormonal sensitivity of the adipocytes. Some of the key hormones secreted by the adipose tissue that could be relevant to asthma include adiponectin. Adiponectin has known anti-inflammatory activity. It promotes insulin sensitivity. It's inversely correlated with BMI. Generally, the higher BMI, the lower adiponectin level a person has. Interestingly, there are adiponectin receptors on the airway epithelial cells. We're not sure what role that has to play there yet. There's also leptin. It's popular because it's the hormone that is responsible for your feelings of hunger. But it's also positively correlated with BMI, and it has a known pro-inflammatory activity. So various studies have shown an association between leptin and activation of specific inflammatory cells or specific immune cells, and it's been found to be expressed in both the airway and airway immune cells. Then there's resistin, which is known for its association with insulin resistance. It's secreted by adipose tissue as well and by circulating immune cells. It also has inflammatory activity. Several studies have surveyed its levels in patients with asthma, but we haven't found a clear trend yet, one way or the other, so we aren't really sure about the specific role that it plays. And then lastly, another important thing is plasminogen activator inhibitor 1, or PAI1. It's another factor that's secreted by the adipose tissue. It's thought to play an important role in adipocyte development, insulin signaling, and also recruitment of inflammatory cells, which could be really key in asthma. It's found at a higher level in persons with obesity, but again, we're not sure the role that it might play in asthma. So it's important to study asthma in those with increased adipose tissue because the data suggests that they may be a unique clinical population. So in this study, we used established guidelines associated with body mass index as a measurement, or BMI, to form our groups. But I want to note as a caveat that BMI is an imperfect measure. We make no assumptions about the specific metabolic capacities of our participants. We're primarily interested in the inflammation and the link to asthma in the microbiome. So we have grouped our subjects to look at their microbiomes and their other asthma features of their asthma. We group them by their BMI. So one of the ways that we assess microbial communities is by measuring and comparing diversity. So diversity asks, who is there and how many are there? We can measure this according to a number of different metrics that have been developed that ask slightly different versions of who's there and how many are there. We can compare the diversity within a single location or a sample, in this case, one person. This is called alpha diversity. Or we can compare the diversity between several locations or several people, which is what we call beta diversity. So if you look at the data here, when we stratified our 16S microbiome data by BMA category, we compared subjects with BMIs over 30, referred to as OB, as those, we compared those to those below 30, which we refer to as non-OB. And we see differences in the beta diversity on the left, according to the unweighted unifact metric, which is a phylogenetic measure of diversity, which means that it can capture information about the groups, how the groups of bacteria that are present in a sample are genetically related to each other. So each point on this plot on the left represents the entire sputum bacterial community of one person. Points that are closer together convey communities that are more similar, and conversely, points that are farther apart convey communities that are less similar. As you can see from the plot, when we colored the points by BMI category, so we have red and blue, the majority of the points from folks with a high BMI, which are blue, are near each other at the top of the plot. So that indicates that there is some sort of association with the beta diversity of these folks based on what group they were in. So we also compared the alpha diversity between subjects in the non-OB and the OB groups. And so although it did not reach our significant threshold statistically, overall, there was generally a higher alpha diversity values in the OB group. So there was associated differences in the sputum bacterial community based on BMI, and this is without even looking at asthma. So just right off the bat, there's differences in the airway microbiomes of folks based on obesity as measured by BMI. So then when we looked at this further, we tested to find what were the specific groups of bacteria that were different in these two groups of folks. And so we had in the OB group, we have higher relative abundance in the airway of bacteria, genre Legionella, some specific Members of Prevotella and Streptococcus were higher in the OB group. In the non-OB group, we had different groups of bacteria that were differentially abundant. So when we further analyzed our groups by evaluating the subjects with asthma, we found some more interesting things, which I'm going to go through here for the sake of time. First, we found there were differences in the sputum microbiota between subjects with asthma and obesity and those with asthma alone. We also found differences in the levels of circulating immune cells between our OB and non-OB subjects who had asthma. Subjects with obesity and asthma had altered levels of some of the blood cytokines and adipokines. So those are the cellular inflammatory signals that kind of set off inflammatory cascades that recruit other immune cells to the area and cause an inflammatory response. So we saw differences in the levels of those signals in the OB versus the non-OB group. And we also saw that blood and sputum cytokines were correlated with clinical measures and measures of bacterial community diversity. So cytokines relay information and they can be part of signaling cascades. As I mentioned, they likely do not exist in isolation. We know that a number of microbes may influence or be influenced by cytokine expression in the body. And so a number of cytokines that we think are important in asthma don't have a defined binary role in terms of whether they are strictly pro-inflammatory or strictly anti-inflammatory or good or bad. Context is likely really important. So we wanted to determine if there were any patterns related to the airway and the microbiome features of interest, in this case, the BMI category and these other cytokine measurements. So our question here is, do the relationships between inflammatory mediators and microbes differ across asthma phenotypes, in this case, in OB asthma and non-OB asthma? So in order to answer a question like this, one of the ways that we can deal with this, where we're talking about relationships within a system, have been pursued through what's called network inference. This type of analysis has been widely used in other fields and has been through a series of fits and starts in its adaptation and usefulness to biological questions. But we're interested in the utility of this kind of analysis to integrate data types and to generate hypotheses about the relationships in a system. So microbiome data have some inherent qualities that make it statistically challenging to infer useful networks, but this is a rapidly developing area that brings together mathematics, computation, and microbiology. One of the most popular types of network applications for biological are correlation-based, but there's lots of development happening in this area. So put simply, the way this works is you just calculate correlations between pairs of components or features like OTUs or specific groups of bacteria that belong to a system. Then those features are then visualized as what is called nodes. They're generally circular. So they're like, you can see the circles here on the right side of the screen. The correlations or associations between the nodes are visualized as lines. These are also called edges. The presence of a line or an edge between the two nodes indicates that there's an association or relationship of some type between them. This could be interactions, it could be a flow of information, it could be a different type of relationship, but it suggests that there is some sort of communication happening between those two nodes. It's important to note that edges do not imply causal relationships. There are also numerous ways that we can cluster the nodes in a network and attempt to determine what the true underlying structure of the components are in a system and which nodes are interacting or associating with each other. In this way, networks are a useful way to visualize potential relationships between multiple parts of a system so that we can make testable hypotheses about them. So we generated networks with cytokine measurements and the microbiome profiles to explore the cytokine microbe relationships in the blood and the sputum we measured. We were especially interested in comparing the OB to the non-OB group. And yeah, that's just saying that networks provide a way to explore data to generate hypotheses, as I mentioned. So here are the networks of sputum microbes and sputum and blood cytokines in each group. I know it's like a rainbow of circles here, but the colors correspond to the clusters that were identified, which remember are areas of potential interaction. Now, there's several things that ways that this network could have organized itself. One, it could be that microbes are only in clusters with other microbes. It could be that cytokines are only in clusters with other cytokines. But I'm most interested in the third possibility, that there are clusters that contain both microbes from the airway and cytokines from the airway or the blood. That would hint that there's some underlying relationship between the inflammatory signals in the sputum or the blood and the microbes from the sputum. So in the non-OB network, there were actually only 2 clusters that contained both microbes and cytokines. Notice in these boxes where I showed you what was actually in those clusters, We have the adipokines, so the cytokines that are secreted by the adipose tissue, and almost all of the other inflammatory markers that we measured are in this one cluster with these 3 groups of bacteria. Then in the second group, we had some other cytokines and then 2 other bacterial groups. And then if we go over to what the OB group looked like, there were several integrated clusters. There was a total of 8, but I've only got 5 here for visibility. There's this orange cluster, which has our adipokines along with a specific type of Prevotella. Then we have this other cluster over here that contained a single cytokine, which is IL-1 beta, and several different bacterial groups of interest to asthma and obesity. So strep, Prevotella, Fusobacterium, a lot of these are bacterial groups that have been found to be elevated in people with asthma and people with obesity. So that was pretty interesting to see that come up. We have lots of other interesting potential associations here that we saw in the data when we did this network clustering. So in summary, we had a number of intriguing findings. First, that the airway microbiota was altered in subjects with obesity without stratifying by asthma diagnosis first. So we had lots of bacterial differences in folks just looking at their BMI without even incorporating asthma. But further, within the subjects with mild asthma, obesity was associated with differences in airway bacterial diversity. So we were seeing further differences in the microbiome in the folks who had asthma along with obesity. So in those with asthma and obesity, we also observed higher levels of circulating immune cells and altered cytokine expression. And then when we looked at our networks, it appears that the immune milieu of blood and airway cytokines and the interactions there may differ between OB and non-OB asthma. So this could have implications for the bacterial communities in the airway, or it might have clinical implications. Maybe there are other ways we need to be looking at these subpopulation of patients in the clinic, thinking about what kind of treatments work for them and why and why not, and what the functions of the bacteria that are differentially associated in those patients, what do they do, what genes are upregulated. These are all questions that I'm pursuing in this project still, so I'm really excited to the follow-up analysis that I'm doing to figure out what the story is here in this group of subjects. So I was only able to share a few highlights of my work with you today, but I'm currently working on several projects that take advantage of the various facets of the data that we've collected, with a major focus on comparing the asthma phenotypes and the impact of ICS treatment on the microbiome. That's one thing that I'm really interested in, to figure out like what's going on in folks who respond to ICS and those who don't respond to ICS. Can we use the microbiome somehow to predict which people are going to respond or not, so we can save them the time and the suffering that they have to go through to try different medications and different inhalers to find one that works for them. That's just one of the projects I'm working on. I'm also working on looking at nutrition and allergic sensitization in folks with mild asthma. And then if you think back to the different major types of asthma, the T2 and non-T2 that I talked about earlier, we also have a project looking at what is going on in the microbiome, trying to incorporate bacterial but also fungal data there to see if there's patterns associated with either phenotype of asthma. So there's lots of exciting things going on in the lab right now. So next, I'm going to talk a bit about the larger implications of microbiology and microbiome research, and particular emphasis that I think will be important in the near future. So microbiomes reflect our exposures. I think I mentioned that earlier. They also reflect our access to resources and services. We know that there are vast inequities in environments and health and access to healthcare, and none of these factors exist in the vacuum. All these things are connected either directly or indirectly. You can see here, this figure is a paper that, a review paper that I wrote a while back, where I talk about how all these things are connected, like the human body within ourselves. We are an ecosystem. We have different microbiomes and different respiratory tracts, the gastrointestinal tract, the urogenital tract. Like there's so many different environmental areas on our bodies that are teeming with microbes that perform important functions for us. We also need to think about the environment, like there are pollutants, differences in climate and moisture, that all of that impacts our microbiome along with our behavior. So the things that we eat, whether or not we smoke, the medications that we're on, the amount of stress that we're under, Also thinking about our proximity to animals, whether they're pets or farm animals or livestock, things of that nature. All of them have a microbiome, and we could be further or closer in proximity to their microbes. We also think about the microbes of our built environment. So, what's floating around in the air in your home, in your community, your neighborhood, in the building. Do you live in a rural area? Do you live in a super urban area? Do you live in a suburban area? There are things, um, um, factors there that also ultimately impact the microbiome and may impact health in a way that's mediated by microbiomes. These are all really important questions that we're going to need to address to be able to address some of the disparities that we're seeing. And it's just really important to bring a more integrated perspective into thinking about the human body and our microbiomes in context of the larger ecosystem that we live in. And so because of that, microbiomes can be a tool, something that we can measure to reflect our exposures, our access or lack of access to resources and services. A perfect example of this has been the COVID-19 pandemic. It was early on, it was clear that COVID-19 was killing people of color at higher rates. It is tempting to assume that this is due to some difference or biological difference in the affected groups that makes them more susceptible. We see a lot of this kind of narrative in the public discourse, but this is not and has never been the answer. When we look deeper into other factors, a clear picture starts to emerge. So for example, as I mentioned, the data shows that people of color who are more likely to die from COVID-19 are also more likely to not have access to testing. They live in high density neighborhoods and housing where it's harder to maintain proper social distance. They're more likely to be exposed to pollution, likely because of proximity to industry and things like that in the places that they live. They're also more likely to have pre-existing condition, and they're also more likely to be an essential worker and not have access to time off and the appropriate quarantine and isolation facilities. And I mean, some people can't afford to take that time off. There are other factors and things like that that have been shown to all contribute to the realities here on the left. A real world example of this from the CDC is a case study where there were massive outbreaks in meat and poultry processing facilities. But 87% of the infected workers were from racial and ethnic minority groups. So all of those things play into this, the figure that we see here on the left. But then when we expand our analysis to examine the context around those cases and trends, other questions present themselves, such as did everyone have access to sick leave in this particular facility? You can see that the CDC guidelines specific to these facilities emphasize the things that could be sources of inequity that could lead to real world inequitable harm on marginalized populations. So COVID is just an example, but in the case of other health conditions and infections and diseases, those of us who are in research and those who would like to become researchers should consider the following things. So what are the problems facing our communities today? We also need to consider who's asking the questions, those research projects, who gets to decide what the research agenda is, what kinds of questions are we asking? What is influencing the questions that are being asked? Who has access to the answers or the solutions? Some of our most critical problems and the problems of inequity that we need to address will require all hands on deck. We need scientists and clinicians from all backgrounds and all perspectives to move biomedicine forward, which means we need to continue to work for representation in science and medicine. So that's one of the things that I have also devoted my career to thus far, is working for advocacy and equity. So this past summer, a friend and I co-founded Black in Microbiology. We had a virtual Black in Microbiology week in late September to celebrate, amplify, and uplift the contributions of Black microbiologists past and present. Black in Micro was a big success, and we have transitioned Black in Micro into a new organization that's called the Black Microbiologists Association. Our mission is to create a community where Black microbiologists can network across career stages and sectors while also supporting the collective work of pursuing equity in the academy, the industry, government, and beyond. Membership is open now. It's free and open to people from undergrad through faculty, both inside and outside of academia. If you're interested, you can join at our website. It's blackinmicrobiology.org. And lastly, I want to dedicate my presentation today to Evelyn Nichol. She was a Black microbiologist and immunologist who contributed so much to microbiology. She was not able to complete her PhD, but nevertheless, she went on to make groundbreaking advances and discoveries. She helped develop the polio vaccine. She was the first to isolate the virus that causes chickenpox in cells. She created a diagnostic test for Toxoplasma gondii, which is an infection that is still in use today. She also has a patent for other technology that is the basis of other molecular diagnostic tools today. Sadly, she died last year as a result of complications from COVID-19, but she retired early from her career in science due to continued racial discrimination and harassment in her workplace, and most of her legacy was lost to history. So I want to thank her family, who I recently connected with, for sharing her story with Black and Micro as we work to gain recognition for her contributions. You can read more about her and other hidden figures in microbiology in a piece that I and the BMA president, Dr. Taylor, wrote for the American Society of Microbiology. There's a link on the slide, and I can send that link to make sure that link gets to you if you're interested in it. So lastly, I want to thank my wonderful co-mentors, my amazing lab members and collaborators for their support, and a special shout out to our study clinical coordinators. It's their job to make sure that the data that we collect from our patients is meticulous. And so I just want to thank them for their work that I depend on. And then lastly, you can find me on Twitter or on my website. Feel free to shoot me an email. I'm always happy to talk to trainees, future and current microbiologists. And thank you for your attention. And I'd love to answer any questions you may have.
Dr. Kozik, thank you so much. That was awesome. Thanks for sharing your story and your research with us. I wish we could all give you a round of applause, but this doesn't allow us to do that. So, but at this time, we'd like to open it up for questions. And there are a couple of ways that you can ask a question. There's a Q&A box that you can hit there and you can type your question in, or there's a hand raise at the bottom of your screen. You can raise your hand and I will open it up so you can ask. I'll put you up, we can hear your voice and even see you on screen. You can ask the question. So, and our people are already sending comments, so thank you for sharing. So our first question is from a student of mine in my class. So I'm proud of my awesome students. So Ebony Walker is going to ask your first question. Let's see if she pops up on the screen here. Okay, there we go. Ebony,
hi, can you hear me?
Yeah.
Okay. Hi, Dr. Kozik. I'm Ebony. I am a super senior here at the UM-Flint. And similar to you, I'm also studying sciences, but I'm going into animal science. So I would like to be a veterinarian. I just find it inspiring to see a woman of color in microbiological sciences. And I was wondering, how do you feel about that? How do you feel about being a Black woman in this type of science?
Well, thanks for your question. And yay, vet school. Purdue, my department was in the vet school. So I have lots of other fabulous Black women veterinarians, now current veterinarians. So props to you. So, I mean, it's really important to me because, I mean, my particular family history has been one where my family has had to fight for the right to an education. My grandfather had to leave the Deep South when he was a boy because his parents were sharecroppers and they wanted the children to be working in the fields, and my grandfather wanted to go to school instead, and they were threatening him and he had to run for his life. And it's one of the reasons how my family ended up in the Chicago area. But so that having that be like part of my story has been something that's been very important to my family education, and pursuing your dreams and microbiology is certainly a field that I love. And so I'm continuously grateful for the sacrifices that my parents and those who came before them made to get us to a point where I can live that and I can do this research and I can do what I'm interested in. And so I hold that very dear each day as I get up to do the work that I love. And I'm just really grateful for the opportunity and the work of those who have gone before me to be able to stand in this space and do work that will contribute to my community, but also the world in general. So I'm really excited to see this movement, specifically in the past year, where there's more recognition of how important representation is and how we need it to solve the problems that we're facing as a society.
Thank you.
Yeah, thank you. Okay, the next question is from Alyssa. If a person was obese, but now is not obese, would they still have similarities of microbiota of an obese person, or would their microbiota change?
That is a really good question. That is very complex. And I think part of what we need to figure out, even in the field of obesity research, which is not really my area, is what does that mean when we say someone is obese or not? Like, because as I mentioned, like BMI is not a perfect index by any means. That's pretty well documented by now, I think. But do you mean simply that they have more adipose tissue on their body? Do you mean there's some sort of metabolic impairment? Do you mean like there's lots of different levels of that? Do you just mean there's systemic inflammation? So I think what happens to the microbiome depends on which of those metrics you're going by, which is something that I'm hoping that the field of endocrine and metabolic, obesity research will kind of move towards being more specific in what specific biological phenomenon they're talking about when we use that word. So I would imagine, though, that the microbiome is going to be responsive to those like subgroups, because inflammation, the microbes and inflammation, they are tightly linked. So like if someone is obese by BMI and then they lose weight and their BMI is smaller, but they still have, if the adipose tissue is just distributed differently, or there's still some sort of chronic inflammation going on, then the microbes are more than likely going to be linked to that, like, deeper level of physiology than they are to just the number on the scale. So it really depends. But that's a really great question. Hopefully we'll be able to answer that soon.
Okay, I have another question here, but I also want to mention that we're going to have a little bit of time left over at the end. And I think we figured out a way that if you want to stick around and have a conversation with Dr. Kozik, we should be able to facilitate that on here. She's going to stick around for a little bit if you'd like to talk directly to her. So the next question is, when it came to analyzing changes in the microbiome of higher BMI patients versus average, were there base changes in the composition of the environment, such as mineral content, pH, water content, vitamin availability, etc.? If so, were these accounted for within the study?
So that's a really good question. And that's something that we are going to follow up on, but it's going to take, I think, a more complex study design. We did collect nutrition information from all of our participants, but our main focus was on the inflammation, the inflammatory phenotypes, and not the obesity angle. So that's something that came up in our data that was very clear that we couldn't ignore. But now that we've seen that, I would love to go back and collect other kinds of information, like you mentioned, which is part of what I kind of talked about at the end. Like, we need to start asking more detailed non-biological, I guess, questions as we are doing these studies, so that we can answer questions like, okay, well, like, is there a difference in the nutrient availability and like the natural resources that these participants have access to? So those are all really important, I think, going to be key questions to really unravel like the relationships at play here. But that's not something that we are equipped to ask in the current study that we have, but we're very much interested in designing more detailed studies to get at those questions.
Okay, thank you. So the next question, do you know what causes the different, do you know what causes the different biodiversity in the microbiomes of different groups, like OB versus non-OB?
Can you say that again?
Do you know what causes the different biodiversity in the microbiomes of different groups?
Okay. So no, we don't know what's at the root of the differences in biodiversity, but we have some ideas. For example, we know that like medication can very strongly impact the diversity of a microbiome. Antibiotics is a big one. So if you give someone antibiotics, their microbial diversity will plummet because you are intentionally killing off a number of microbes. So there's less variability among the microbes that are there. Whether or not there are lifestyle or behavioral factors at play in the OB versus non-OB groups, that's something that we're going back through our patient demographic data to figure that out. If there's a clear variable or something that could explain that difference. That's also one of the focuses of our study on ICS, because we don't really know what happens when someone inhales a steroid. Are you changing the environment so that only certain kinds of microbes can grow? Because if that's the case, then you're going to lose some diversity there because you're selecting for the ones that can, whether it's metabolize the drug or aren't affected by the drug. Those are questions that we're also pursuing because it could be any number of things. It could also be diet. I mean, this is more established in the gut microbiome literature. It's really new to think about this in the context of the airway, because up until, as I said, 10 years ago, they didn't believe there was any microbes in the lungs unless you were very, very ill. So we're starting to piece together like, okay, does your diet affect the microbes in your airway as well as the ones in the gut? Because it could be that those are linked to dietary or nutritional differences. Those are also things that we're pursuing with our data.
Okay, thank you. The next question is from Mary. Hi, Mary. Do you know if there's a correlation between other diseases that cause chronic inflammation and the microbiome? For example, inflammatory diseases like heart diseases.
Right, so I would say yes. So we know that inflammatory diseases in particular seem to show a difference in the microbiomes of people who have those diseases. So as I mentioned, in grad school, I worked on IBD. So that includes Crohn's disease and ulcerative colitis. So in patients who have either of those diseases, where there's like a lot of gastrointestinal inflammation, they do have very different microbiome compositions. And that's one of the things that I think is really cool about microbiomes and inflammation is they're so tightly linked to each other. Like when you have an inflammatory process, there's a lot of inflammatory signals and cytokines that are being sent everywhere. And the immune cells are congregating, responding to those symptoms. But because of that, you could have like changes in the level of mucus, for example. So you have people with inflammation, there's increased mucus in their gastrointestinal tract. So that gives more nutrients for microbes to eat. So then you're going to grow different kinds of microbes there that can use the resources that are now suddenly available. And so I do think that there is a link in other inflammatory diseases. Another one is arthritis that comes to mind, like inflammation of the joints. We see differences in the microbiome there. So it's really, it's going to be really key for us to figure out what the relationship is between the microbes and inflammation in all these different conditions.
Okay, does anybody have any other questions? That's the end of my question list. Either raise your hand or send a question. Double check. Well, you've had a lot of thank yous in the chat. Oh, wait, there's another one. Follow-up question to the biodiversity question. Do antibiotics cause permanent changes to a person's microbiome, or does it recover eventually? That's a great question, and also one of my questions.
So yeah, so there's some really cool work being done about this by one of my colleagues here, Pat Schloss and his lab. They've done a lot of, and I think Vince Young. They've done a lot of work with antibiotics because they work on people who have Clostridium difficile infection, which is a really nasty gastrointestinal infection where you have an overgrowth of one particular bacterium. And usually those people have to go through really heavy antibiotic treatment to get that infection to resolve. And so they have found that there is significant alteration of the microbiomes, but in some cases there is a recovery that happens. It may not be the same amount of time for everyone, and they're in the process of trying to figure out whether that recovery is back to where it was before, or if you have like specific changes in what a new normal would be for that patient. And I think that's one of those things that is going to depend on other factors also, like is the person's diet modified, like, you know, after like the infection, or is there going to be some follow-up medication that they need to be on that will continue to exert some sort of environmental pressure on the microbes there. But it's one of the reasons why right now in medicine, there's like a push to kind of be more conservative in our antibiotic use, not only because of, I mean, the effect of the microbiome, but also that like the microbes themselves are becoming resistant to the antibiotics. So we don't want to, you know, throw everything at everything if we don't need to, because it could be having like ecological impacts that we weren't anticipating. Right.